Hello, APBIO. In this last bit of notes, I'm going to talk about prokaryotic gene controls.
One of the main mechanisms of the control of gene expression is a mechanism called an operon. And the first thing I want to do is let's define an operon, and then let's talk about what the components of the operon are, and then I'll talk to you about how it works. So we kind of need to get that out of the way first. So the first thing let's do is define it. Again, just to stress, we're talking about gene control in prokaryotic cells, not eukaryotic cells. The operon is a cluster of functionally related genes. So genes that are all located next to each other on the DNA, one adjacent to the next, grouped into a single transcription unit, which means that an RNA polymerase can come down and transcribe all of them at once. And all of the genes in that transcription unit are going to be controlled by a single promoter. So remember that a promoter, by a single promoter, that's where RNA polymerase binds, and something called an operator. And an operator is going to act as a switch. We're going to talk about that in a second. So you can remember all the parts of an operon with the acronym PROG, but not spelled like the city. It's spelled like frog. And it's PROG2. Okay, so let's go over what each one of those letters mean. The first letter, P, refers to the promoter. And the promoter, we've talked about it before when we talked about transcription. That's just the DNA sequence where the RNA polymerase binds. And we have a promoter on eukaryotic genes too. All genes have a promoter. It's just the binding site for RNA polymerase. The R stands for a repressor protein. So this is a protein, not the DNA. And it's a protein that regulates, so we call it a regulatory protein. And it acts as to block transcription. So that's why it's called a repressor protein. And it binds to a region on the DNA called the operator. And the operator is like a switch. And when it's bound, it blocks transcription. Like I said, the operator is where the repressor protein binds. So this is also a DNA sequence. But this is not where RNA polymerase binds. This is where the repressor protein binds. And the operator acts as kind of an on-off switch. So let's put that in quotes. On-off switch. It's going to be able to turn on off the gene with the help of the genes with the help of the repressor protein. So we have the promoter, the repressor protein, the operator, and now we have G2. There's two genes. That's why it's G2, two types of genes. The structural genes we'll talk about first. That's really the purpose of the operon. So those genes are code for functionally related proteins. Code for functionally related proteins. They're all grouped together, adjacent to one another, and they're just proteins that are needed for proper cell function. And I'll give you some examples of some different operons, and the operons are typically named for the structural genes. So we'll talk about the LAC operon, the ERA operon, the TRIP operon, and they are just going to code for these functionally related genes, the structural genes. Okay, now let's talk about the regulatory genes. The regulatory genes are, they're going to code for the repressor protein. So they code for this guy right here. Code for repressor protein. And they're called regulatory genes because that repressor protein is turning on and off. It's regulating transcription. And they're actually located upstream of the operon. So they're not considered an actual part of the operon, but they're a part of the mechanism. Okay, upstream of the operon. Let's look at the different parts of the operon so you can see what it looks like. So if you can imagine, we're talking about a chromosome, a prokaryotic chromosome, so it's a circular chromosome, and we're talking about a section of the chromosome. So an operon, this is inside a cell, right? And the operon would be just one section, one little section right here, say. That's the operon. And let's talk about how we can place all the different parts on the operon. Here we go. This right here is, let's label all the different parts. Here's RNA polymerase. And if that's RNA polymerase, we know here, this orange thing right here is thinking about where RNA polymerase binds. Remember, that's the promoter. And then we have, this is that repressor protein. Right there. And we talked about the repressor protein binding to the operator. That's that on-off switch. These right here are the genes that are located, functionally related genes that are located one next to each other. So these are the structural genes. And then we'll show a break right here and we'll draw in. We don't have it located here, but shown on this diagram, but this right here, this would be the regulatory genes. Which means that these regulatory genes right here would code for this repressor protein right here. Or the green, showing the green. Now, if we look at this particular diagram right here, when the repressor protein is bound, you notice how it's physically just blocking RNA polymerase. So this is showing the operon in the off position. Here it is in the off. Now, let's look down and look at how the operon works. In this particular case, we have this white little circle right here is called an inducer. And the inducer is going to pull the repressor protein off. Here's the repressor protein. So there's something in the environment, and we'll talk about what that might be, that is pulling the repressor protein off. Now, when the repressor protein is off, here's RNA polymerase. And here, right here, is the promoter. And you can see how RNA polymerase can just slide right past the operator, a little red sequence right there, and transcribe the structural genes because the inducer has pulled it off. So this is in the on position, and we say that the inducer has induced the transcription of whatever the structural genes are. Okay, let me give you an example of an operon that works like the one we just labeled. The lac operon is what we call an inducible operon because it works just like this diagram. So let's talk about that for specifically. So the lac operon, its job is to code for enzymes that catabolize lactose. And if we think lactose is a disaccharide, so here's lactose right here. Let's just go A, B. And in order to use lactose, you need enzymes to break it down, or bacteria need enzymes. They're the enzymes, and they're going to break it down into its parts, to its monosaccharides. Oh my gosh, excuse me. And like I said, it's a disaccharide composed of galactose, which is a monosaccharide, and glucose. And so this right here, these enzymes are needed. Now, there's multiple enzymes that are needed. And so on the lac operon, we've got 3 different enzymes, 3 different genes that code for the enzymes that break apart lactose so it can be used. Okay, so what happens to the repressor protein in the presence of lactose? So when lactose is present, what happens is normally that repressor protein, which is produced by the regulatory genes, is going to be bound to the DNA. And if there's no lactose in the environment, it doesn't make sense for the bacteria to produce the enzymes that are going to catabolize lactose. But if lactose is present in the environment, then what's going to happen is then the cell can use the lactose to break down into these monosaccharides, and then it can make ATP with that. So it can use an alternative food source other than glucose. Well, that's good, right? And so it allows the bacteria cell to respond to the environment. Now, when lactose is in the environment, the repressor protein is going to be released, because let's imagine that this right here, in the inducer molecule, in the case of the lac operon, is lactose. So the substance that actually is going to be catabolized actually turns on transcription. That's what is called an inducible operon. So the presence of a particular substance is going to turn on the operon. So let's write a few things down. The repressor protein, when lactose is in the environment, the repressor protein is deactivated and then released from the operator. So essentially, lactose is the inducer. Not essentially. Lactose is the inducer. Now, because lactose is released, then RNA polymerase can bind to the promoter and transcribe the structural genes. In other words, the genes are expressed. And because the genes are expressed, then lactose can be broken down. And again, we call that an inducible operon. Now, when the repressor protein, when the lactose is all quote unquote digested or catabolized, and there's no more lactose in the environment, look what happens. No more lactose in the environment. Then the repressor protein is reactivated or activated, and the repressor protein binds to the operator. And turns off the operon. So you can't digest lactose when lactose isn't present in the environment. Well, that makes sense, right? We call the lac operon an inducible operon because it's turned on in the presence of a specific molecule. In the case of the lac operon, lactose. So all of the operons are going to be turned on specifically by the presence of the material that they act upon. Another inducible operon is the arabinose operon. And so I want you to pause the video right now and I want you to read about the arabinose operon. Typically, we would do an investigation that involved the arabinose operon. I think this year we may not do the investigation, but we are still going to learn about the arabinose operon. It's an important concept. And then after you read about the arabinose operon, answer question number 9. And then I want you to read about using the operon in order to engineer gene control. And we'll talk a little bit about this in class. We're going to talk about the green fluorescent protein and engineering DNA. So just read this right here. And we're going to talk about engineering that DNA. And then you can keep watching the video.
Many operons are also repressible operons. So they're in the on position, essentially, as naked DNA. And then they can be turned off in the presence of something. And that's what the trip operon is. So it's called the trip operon, the TRP operon. So I'm here in your notes. And the purpose of the trip operon is to code for enzymes needed for the synthesis of tryptophan. So if you can recall from our conversation in biochemistry, and then again looking at protein synthesis, tryptophan is one of the 21 amino acids. So organisms need it to be able to make proteins. It's not an essential amino acid for bacteria. They can actually make it. But if it's in the environment, it doesn't make any sense to make it, right? So this question number 11 asks, what is tryptophan? Why do organisms need it? You could answer that question. And then it's tryptophan and the trip operon is a repressible operon. So it's opposite of an inducible operon. So let's think about what happens. The repressible operons here, so let's orient ourselves. Here we have the promoter. Remember, that's the operator. Here's the operon right here. Here's the promoter. That's where the RNA polymerase is going to bind. And then here's the repressor protein, this pink thing right here, that's bound to the operator. And then here's all the structural genes. So if you notice, in the presence of tryptophan, so in this case, these are repressors. This specific small molecule, in this case, because it's the trip operon, it's tryptophan. So this amino acid binds to the repressor protein, and it activates the repressor protein while it's bound. So before, with the lac operon and the arabinose operon, remember, in the presence of arabinose, this pulled off or allowed for the binding of RNA polymerase in the presence of arabinose. But in the repressible operons, the presence of a molecule is going to inhibit the binding of an RNA polymerase. And you can see this guy here is trying to get on and bind to the promoter, but it can't because the trip repressor is active. The repressor protein is active. Now, when tryptophan is low, this trip repressor protein, the trip repressor is going to be inactivated, meaning it's going to pull the repressor protein off of the operator, and now our RNA polymerase can bind and transcribe the genes. So it acts the opposite. So if we think about how the regulatory gene is synthesizing this repressor protein, in repressible operons, the repressor protein is synthesized in the inactive form. It's not active. The only way it's activated is in the presence of something else. So this molecule here activates and inactivates this protein, right? That's the opposite from inducible operons. In inducible operons, let's go back and just look quickly at the lac operon. In the lac operon, it's inactivated in the presence of something. So it's inactivated in the presence of something, and here it's activated in the presence of something. So let's go back and look again. This is inactivated in the presence of something. So it's pulling it off in the presence of something. And this is making it stay on in the presence of something. So the mechanism is opposite. So in repressible operons, the synthesis of the repressor protein is in the inactive form. But in inducible operon, the synthesis of the repressor protein is in the active form. You see, this is the active repressor, and it binds directly when in the absence of something, but it is induced when the specific small molecule is in the environment. So in the repressor protein, in the presence of tryptophan, is going to be activated, because why would you make tryptophan if you already have it? That's silliness, right? And in the absence of the repressor protein, in the absence of tryptophan, is activated. It comes off of the DNA, and it is. It's inactivated. It comes off of the DNA, and now RNA polymerase can transcribe those genes, bind to the promoter, transcribe the genes, and then make tryptophan so that those bacteria cells can then make proteins that have tryptophan in them. Okay, that's it, and I hope that was helpful.